Okay, so now we're going to be looking into the steps of the Calvin cycle. So to begin the Calvin cycle, we have ribulose, 1,5-bis-phosphate. And just to save time, I'm not going to draw the structures. I'm just going to draw the names and the steps. So ribulose 1,5-bis-phosphate can go through two steps. It can go through carboxylation, which involves the input of the carboxylase enzyme, and it also goes through oxygenation, which is through the input of oxygenase. So it's really important to note that these two steps are irreversible, and they are an exergonic process because they don't require the input of energy, yet they're irreversible. And that creates 3PG, and for the oxygenase, it creates 3PG, but also 2PG. So now this enzyme, the carboxylase enzyme, is actually the, I'll write it right here, the rate-limiting step of the cycle. And note that it is highly exergonic and irreversible. Okay. Also note that Rubisco is the only enzyme that enables the fixation of CO2 for the formation of biomass. And it is found in both plants and cyanobacteria, so it's not exclusive to only bacteria or plants that only use oxygen for energy. Okay, so now let's go into the process of what you guys need to know. So most likely you won't need to know how to draw the entire pathway, most likely just the fixation and the reduction step, but not the regeneration. So the process begins with the breakdown of 3PG. So you add an ATP and you get out a DP. This is also this is reversible, right? So make sure you're keeping note of all your irreversible steps. And that gives us 1,3-bisphosphate glycerate. Okay, and note that in this specific structure, we have a mixed anhydride plus the phosphodiester bond. The mixed anhydride is a higher energy than the phosphodiester bond. I'll draw where the mixed anhydride is. So I'll draw the structure for this one. So here is the mixed anhydride, and here is the phosphodiester bond. So what makes this a mixed anhydride bond is the fact that it's between two high energy structures. So you have like the carbonyl plus the phosphate, whereas this one is just a carbon and a phosphate. Okay, so after you create your 1,3-bisphosphate glycerate, you have the input of NADPH, which makes NADP+. So we oxidize the NADPH, and we removed a phosphate here. So we have D-glyceraldehyde 3-phosphate. So we lost a phosphate, right? Because we went from having a phosphate on position 1 and 3, and now we have the phosphate only on position 3. And this structure can use the triose phosphate isomerase, and that will actually make it a dihydroxyacetone phosphate. Okay, so before we move on, I just want to know a couple of important processes. Also, let me highlight these so that way, I'm going to highlight the products so that way it looks a little more cleaner. Okay, and then I'm just going to add in the enzymes in green. So here you're adding a phosphate, so you're going to need some type of kinase enzyme, and it's called, for this reversible step, it's called Phosphoglycerate Kinase. For this one, right, we talked about this in a previous question. Anytime you have oxidation or reduction of an energy carrier, then we have dehydrogenase. So this one is NADP glycerine aldehyde phosphate dehydrogenase. So we oxidized NADPH, therefore we reduced 1,3-bisphosphate glycerate. And then this final step is through the triose phosphate isomerase. Okay, something I wanted to note as well is that the kinase step here and the dehydrogenase step, these are both endergonic, because energy is released by the broken bonds from the structures. Okay. So also, you do have a high energy phosphoanhydride bond broken, and you have a new bond that's favored. So you didn't lose any energy, which is why your step here is still irreversible. Okay. So next, we can actually break this down even further. So we can use aldolase enzyme, and we make fructose 1,6-bisphosphate. And then we can use, I'm going to highlight these enzymes in green so we don't lose them. So then we have bisphosphatase enzyme. And note, this is an irreversible step. So knowing the 4, I believe it's 4, 1, 2, 3, 4 irreversible steps for this process. So we have bisphosphatase, which makes fructose 6P, and that can switch into erythrose 4P, and then using an aldolase, we make cetoheptulose 1,7-bisphosphate. Okay, another thing I wanted to note is that the D-glyceraldehyde 3-phosphate can actually switch into a different type of sugar through enzyme transketalase. And it can become Cellulose 5P. So I'm just going to highlight these products so we don't lose them. Okay. And then this acetoheptulose 1,7-bisphosphate can use bisphosphatase again. Again, this is an irreversible step, so make sure you keep note of those. And that gives acetoheptulose 7P. Okay, and then that can move through using transketalase. And that can make Cellulose 5P. So technically, you could go through this entire process to make Cellulose 5P, or you can use transketalase using the D-glyceraldehyde 3-phosphate to make the Cellulose 5P. Okay, so you will most likely be asked to draw the fixation step. I think that's the most important that Dr. Thomas talked about. And just like understanding where the phosphoester bonds are, the mixed anhydride bonds, knowing your irreversible steps. Another thing is knowing that you can switch between the keto form. This is a resonance. I'm using resonance arrows, I'm sorry. Through tautomerization, use the enol form. I mean, technically this is, it's just like resonance, because you're moving the electrons around, and here you have a keto, right? So you have like two R groups, versus here you have an enol. Okay. So make sure you also know all the irreversible steps. Okay, so we're going to move on to the different types of carbohydrates. Make sure you know that sucrose is made from glucose plus fructose, and that you have a 1,2 glycosidic linkage between the two. Okay, so let's start with starch. Okay, so starch, you want to know that it is a 1,4 alpha glycosidic linkage and some 1,6 alpha links. Okay, it also has two subtypes, which are the amylose and amylopectin, which we'll also get into in more details. And it's the storage form of high energy carbohydrates in plants. Okay, so these I think are the most key important parts or characteristics of starch. So now let's get into amylose. So amylose is linear. When something is linear, it's not branched. And it's a 1,4 alpha glycosidic link. And that's honestly the most important part that you need to know. And it's just an 1,4 alpha glycosidic linkage of linear chains chains of glucose. So there's no branching. Okay, and then you have amylopectin. Amylopectin, unlike amylose, is highly branched. Okay, remember from your glycogen pathway, it's highly branched. And it contains 1,4-alpha plus 1,6-alpha links, which is also how it's different from amylose. So it kind of looks like, so if I had, if I have this glucose molecule, so I have my O link here, and it's being attached to another, my chair structures are very ugly, so I apologize for that. But it looks like this, whereas this might be linear because you just have like the whole chains being linear, but then you might have like this part right here, which makes it branched. Okay, finally we have cellulose. There's a little more you need to know about cellulose. It is a polybeta glucoside linked at C4. It's largely linear, and it's the main component of plant cell wall. And you also have this enzyme called cellulase, which catalyzes the hydrolytic breakdown of cellulose. So cellulose, humans don't have cellulases, so that's why like you hear corn, for example, we can't break down corn because it contains cellulose. We don't have that enzyme to catalyze the breakdown of cellulose, therefore it's not digested in our bodies. So it does not exist in animals, but it does in some fungi and bacteria. Okay. Also, I forgot to mention, but so we do have these two pathways of carboxylase and oxygenase for the rubisco. So this is rubisco. And oxygenase is actually highly problematic because it causes the loss of energy. And as you can see, you go through this entire process and you do lose a lot of energy that way. So just keep that in mind as well. Okay.